Hi everyone. In today's lesson, we're going to look at detecting genetic disorders. There's going to be two different methods that we'll look at. Both methods are fairly accurate, and you can do them at different times, which is why there are two methods. But there are some similarities between the two as well. So the first one that we're going to look at is called amniotic fluid test, or AFT. Now, the old name for this is amniocentesis, which is something that your parents or grandparents would recognize more. But today, we simply call it AFT, or amniotic fluid test. The second detection method is called chorionic villus sampling. Chorionic villus sampling can be abbreviated as CVS. And this one here, again, it's kind of similar to the AFT, but it takes your cells from different locations, okay? And depending on where it's being taken, okay, again, you can make a better karyotype to determine whether or not your child, or future child here in this case, has any kind of chromosome disorder or not. So these two methods, their main goal is to obtain a tissue sample or a cell sample, okay, of the fetus. Okay. And those cell samples are then used for karyotyping, which is, again, a process that we've looked at before, where you can see all the different types of chromosomes that are in each one of the cells of this individual. Okay. And we look for abnormalities in their chromosome number. So AFT, by now you can kind of see that it involves a device called an ultrasound. An ultrasound scanner basically emits a bunch of sound waves, and those sound waves is able to go through skin and muscle. Okay, it bounces off skin and muscle back into the scanner, where a computer basically then analyzes the data and generates an image, okay, of what it quote unquote sees. Okay, so it's useful because, well, if you look on the left hand side here, this person is sticking a needle into the womb. Okay, this needle here is used to extract something called amniotic fluid. This is basically the nutrient sac that surrounds the baby, okay, or the fetus, and it's filled with fluid and nourishment. It also acts as a place for waste products to be deposited as well. So when the baby is growing, okay, when this embryo grows into a fetus, eventually some of its skin cells will slough off into the amniotic fluid. And that's what we're trying to extract. We're trying to extract some of the cells that have been sloughed off and deposited into the amniotic fluid itself. So your amniotic fluid thus contains cells from the fetus, and from that we can use it to make a karyotype. The benefit of an AFT is that you can do this between the 14th to 20th week of pregnancy. So this is still pretty early on, okay, and it's early enough for you to be able to detect several things from the karyotype. Okay, you can detect gender at this point. Okay, even from the ultrasound, you should be able to make out the gender of the individual, because usually gender has been determined about the 7th to 10th week of pregnancy. So by the 14th, it's definitely there. Now, I'm just going to move away so you can see this bit. Move over here. Okay. So like I said, it does use ultrasound and a needle to extract that amniotic fluid. Now, some of you guys are probably wondering, well, what happens if you poke too hard, or if you insert the needle incorrectly? There is a slim chance, okay, of the baby moving sometimes, but for the most part, there is enough room inside the amniotic sac so that you would not injure the baby. Okay. Also, that needle will puncture and make a very, very tiny hole in the amniotic sac, which doesn't end up leaking out. Okay, it does actually get repaired, and the fluid that you extracted from will most likely be replenished within 24 to 48 hours after you've extracted it. So there's actually very little harm that can be done to the baby. So this is what I was talking about, the amniotic sac. You can see the umbilical cord that's attached, okay, and it'll be attached to placenta, but the sac is just a very, very thin layer of membrane, okay, but it's filled with fluid. So when the needle goes in, it makes a tiny little hole, it sucks up a little bit of fluid, and then it pulls out, okay. And because the hole that it makes is very small, okay, there's very little amniotic fluid that will leak out of it, okay, and basically this sac will just repair itself, okay, in time. This is a great test if you have concerns about birth defects of your child, okay. So we usually do use it to test for trisomy or monosomy disorders, okay, or basically non-disjunction disorders. So it's not something that every doctor will perform on the expecting mother, and it is usually recommended from doctors. Okay, so if a doctor feels like there's a need for it, they'll ask you to do an AFT, or if you have a family history of genetic disorders, then you can also ask the doctor to recommend AFT being done. Okay. The next one is called CVS, so chorionic villus sampling. This one, again, also uses an ultrasound, but it doesn't use the needle. Okay. Instead, they will take a catheter, which is a long, thin tube, and they're basically going to push it through the vaginal opening so that they can get into the womb and then extract a piece of tissue, okay, from the actual placenta. So that's why this technique is a little bit more invasive, okay, than just sticking a needle in and extracting some fluid. But the benefit of this particular method is that you can do it a little bit earlier than an AFT. Okay, so it can be done as early as 10 weeks, but usually around the 13th week of pregnancy. So again, it's not significantly early, okay, but for some people, they probably need to know earlier if they have strong suspicions that something's wrong. So they'll use ultrasound in the same way, okay, just to detect where that fetus is. The catheter itself, it doesn't hurt as much as putting a needle in, but it is a little bit intrusive because it's going through a very sensitive area. But eventually, once you extract a tissue, that tissue is then again used to create a karyotype. This is definitely recommended for mothers that are 35 years of age or older. Okay, so usually the reason for that is when you have children at a later age, there's a higher risk of chromosome disorders, okay, or complications. So they want to make sure that you're aware of the condition of your child, okay, and the health of your child. So it says that it's commonly done for trisomy 21, but once you have prepared that karyotype, you could probably find out any other information, okay, like if your child happens to have trisomy 18 or trisomy 13, or even, you know, Cretoshat, right? So any of your chromosome disorders, whether it's autosomal or sex chromosome based, just because you have the karyotype, you can diagnose those as well. Okay. Now this one I will post as a video. Actually, there's several videos that were embedded in this lesson here that I didn't play, but I will post it, a link to it, so that you can at least watch them. I actually like this particular video because it shows more or less a time lapse of how a baby develops from being an egg fertilized by a sperm all the way up to birth. Unfortunately, it's not like a real life video of a child being, conceived and born, but it's actually a pretty interesting one. So I'll make sure that I post this one as long as the link is fine. So to summarize, when we're trying to detect genetic disorders, our current technology that is common is AFT and CVS. And both of them will perform their jobs accurately, okay, because both are just means of getting cell samples from the actual fetus or embryo. So once you have that cell sample, it's all basically making a karyotype and then analyzing that karyotype to see if your child has any genetic disorders. That's all for me. See you guys next time.